Now we've completed the second section of this lecture. We're going to go to the third section. We have at this time defined translational research. We've provided a functional understanding of the homing process. We've described the molecular effectors of homing to the marrow. And now we're going to focus on how we could achieve vascular delivery of stem cells by glycosyltransferase program stereosubstitution. The abbreviation for that is GPS appropriately, and utilization of a chemokine bypass pathway that we've discovered in our laboratory. So just to review some important definitions. Glycans, by definition, are multi-sugar structures. Receptors and ligands they function as locks and keys. Doesn't matter whether you call something a receptor and its co-receptor a ligand or vice versa. Just recognize that one end of a molecular pair will be a receptor, the other end will be a ligand. And glycosyltransferases, the enzymes that modify scaffold, protein, or lipids to create carbohydrate decorations. Those are the glycosyltransferases. So GPS stands for glycosyltransferase programmed stereosubstitution of cell surface glycans. It's a technology we developed in our laboratory to allow us to modify the surface of living cells without affecting cell viability or function. Now, within the Golgi apparatus, it is known that the creation of a carbohydrate modification occurs in an assembly line fashion where monosaccharide substitutions occur on restricted acceptor sequences, such that the process of placing ultimately a sialic acid on the terminal lactosamine requires first that a galactose be linked to an N-acetylglucosamine, and thereafter the sialic acid will be placed on that terminal galactose, so that these things occur in sequences. And not to simplify the process, because it's very complicated. At the same time, there may be glycosidases in this milieu which are trimming back molecules that were already modified so that only certain sequences will ultimately contain the monosaccharides that would be dictated by that glycosyltransferase. Nonetheless, this is typically how it occurs inside the Golgi and starts off, in fact, in the endoplasmic reticulum. GPS, on the other hand, seeks to recapitulate that biology by placing the relevant monosaccharide substitution or substitutions on the surface of the cell, on the surface of the carbohydrates already present on that cell surface. So in the case of GPS to create H cell, we identified using monoclonal antibodies, principally the monoclonal antibody known as SAC1, that the CD44 glycoform expressed on a mesenchymal stem cell actually had a sialylated lactosamine terminus. That is a sialic acid alpha-2,3 linked to galactose, beta-1,4 linked to N-acetylglucosamine, a type 2 sialylated lactosamine backbone. And that the only thing that was missing to complete the sialylated Lewis X motif was the fucose substitution in alpha-1,3 linkage to the N-acetylglucosamine. So we sought to create an enzyme, in this case the fucosyltransferase 6, that would modify that N-acetylglucosamine specifically with an alpha-1,3 linkage of the fucose, and do so without affecting cell viability or native cellular phenotype, to allow then just the modification to exist and then seek whether that possible modification would allow the cell to traffic into the marrow. As we perform this modification, we would identify whether H cell was created by performing Western blot of CD44, looking for reactivity with E-select and Ig chimera, or with monoclonal antibodies KM93 and HECA452. So shown in a different cartoon fashion here is essentially what was done, simplified, so that we recognize that the CD44 molecule had N-linked, importantly, N-linked carbohydrate substitutions that had sialylated lactosamine termini that had not been fucosylated. Then, by using the fucosyltransferase enzyme in combination with the sugar donor GDP fucose, we could then fucosylate these termini with alpha-1,3 fucose substitutions and create sialylated Lewis X. This is all reviewed in a major review article written in 2009 in Immunological Reviews, which I point to your attention if you want to hear more about this biology and chemistry. Now, for the guiding principles of GPS, I want to point out some important points. Number one, to perform this properly and elegantly, you must first identify the relevant glycoconjugate acceptor that you seek to modify. the protein and or lipid carrier that you want to modify with a specific carbohydrate substitution. This can be accomplished by using monoclonal antibodies in flow cytometry combined with blotting. In the case of a protein, you would do a Western blot. In the case of a lipid, you do Far Eastern blotting. You then use pertinent enzymatic conditions and reagents, properly formulated, importantly, properly formulated to avoid affecting cell viability and phenotype, and then confirm cell viability and functional status after completion of the GPS reaction. Again, confirming cell viability, an important part of this process, and the functional status of the cells. And then to verify that modification of the intended scaffold, either protein or lipid, by biochemical and functional assays, including appropriate in vivo assessment of the desired phenotypic effects. So now shown here is a diagram from a report we had in Nature Medicine in 2008 describing this biology and this application to mesenchymal stem cell homing. And in panel A here is the flow cytometry of a variety of different probes, HECA 452, KM93, and E-select and IG Chimera, before and after the GPS reaction. And in every case, you can see by the shift in mean channel fluorescence a marked increase in reactivity with HECA 452, with KM93, and with E-select and IG Chimera, indicating that we created the sialylated Lewis X molecule and that it was obviously functioning in E-select and binding. Importantly and remarkably, the only scaffold that expressed the HECA 452 reactivity after the GPS treatment was CD44. The only molecule on the surface of the mesenchymal stem cell that displayed the relevant acceptor sequences to have ultimately sialylated Lewis X created by fucosylation was CD44. We were quite surprised by that because we had reasoned that there might be other acceptor molecules that were under fucosylated that might be sialylated Lewis X bearing after the GPS reaction. But in fact, the only molecule that was poised to display sialylated Lewis X was CD44. And that's shown here because by Western blot, the only molecule that reacted after the GPS fucosylation with FT6, the only molecule that reacted with HECA 452 was CD44. And down in the lower panel, we showed biochemical evidence that it was, again, only CD44 because we immunoprecipitated the molecule. It was EIG reactive. It was HECA 452 reactive. If we treated the molecule with N-glycanase to remove N-glycans, the reactivity went away, confirming that the target scaffold carbohydrate was an N-linked glycan, and that this molecule, the CD44 molecule expression, did not change with regards to the fucosylation reaction or treatment with N-glycosidase F, indicating that the core molecule itself is not affected by the GPS reaction. The conclusion of all this is that fucosylation of mesenchymal stem cells, human mesenchymal stem cells, creates H-cell, and that CD44 is the only mesenchymal stem cell protein that displays sialylated Lewis X. Now, to observe and to analyze whether the GPS reaction was effective in driving the homing of a mesenchymal stem cell into the marrow, we used immunoincompetent mice, immunosuppressed mice, the Nodskid model of mouse, which lacks the capacity to reject human cells, and introduced the mesenchymal stem cells in the mice in combination with intravital microscopy. So we placed a 2-photon microscope on the surface and a laser microscope on the surface of the calvarium and studied the actual homing of the mesenchymal stem cells introduced into the mouse in real time. To do this, we dye labeled the cells with fluorescent dyes, and if you look over here on the right panel, within 1 hour of injection of the cells, we were able to obtain evidence that the cells were actually aggregating or collecting within the sinusoidal endothelium of the mouse calvarium. Within 24 hours, we could find, coming back to the very same vessel, the extravasation of those cells, whereas at 1 hour they were present within the lumen of the vessel. At 24 hours, they'd already exited the vessel and were into the parenchyma. And if we came back 12 days later by 2-photon microscopy, we could see the cells lining up against the endosteal surface of the bone marrow, which is where bone is formed in a mouse and in a human. And these cells were then localizing to the proper loci for bone formation in the mouse bone tissue. So human cells introduced into the mouse were being driven to migrate into the marrow and were thereafter partitioning within the marrow microenvironment in the spot where bone would be created. If we came in 6 weeks later and performed immunohistochemistry of sections of the mouse calvarium and other bones, we could clearly see the production of human bone in the mouse bone. Shown here are a series of panels, the first of which is just a light microscopic picture of the sections. The next panel is staining for human CD44. Down here you can see where the cells that had been modified to express H-cell were injected into the mouse. They had aligned against the endosteal border of the intramedullary cavity, and these cells, CD44 positive cells, were making human osteocalcin. Next panel over is the staining for the human osteocalcin protein. And if you merge these figures, you could see the yellow where the human CD44, that is the human mesenchymal stem cells, were producing human osteoid in the mouse marrow. In the case of mesenchymal stem cells that were injected that were not modified by GPS to express H-cell, we didn't see evidence of accumulation of the cells in the marrow whatsoever, and there was certainly no production of human osteocalcin in those mice. So therefore, the ability to target trafficking of the mesenchymal stem cell into the mouse marrow was dependent on the enforced expression of H-cell via GPS. Now, how does this work? Well, the carbohydrate substitution only lasts 24 to 48 hours. The CD44 molecule has a surface turnover of that half-life of about 24 hours, so that the molecule only functions in the initial capability to direct trafficking of the cell into the marrow. Once the cell has transmigrated into the marrow parenchyma, the H-cell molecule is turned over back to the native CD44 structural state. And that CD44 molecule then seeks out hyaluronic acid, which is deposited on the endosteal surface of the intramedullary cavity, which is where bone formation occurs. And the cell then will differentiate into an osteoblast and make the osteoid in that section of the bone. Now, critically, one of the issues that was left for us to figure out here was that we identified that human mesenchymal stem cells generally lack chemokine receptors. Earlier in this presentation, I talked about the critical role of SDF1 binding to chemokine receptor CXCR4 on hematopoietic stem cells. But mesenchymal stem cells lack CXCR4 expression. So therefore, how was it that a cell that had enforced H-cell expression was able to gain access into the marrow without chemokine input? That was an important question for us to determine an answer to. And critically, though, remember that the mesenchymal stem cell expresses VLA4. So it has the effector of the step 1 cascade if you enforce E-selectin binding, that is H-cell, and it also possesses VLA4. So missing was the centerpiece, how this activation of VLA4 occurs. Through subsequent studies in our laboratory, we identified that the human mesenchymal stem cell, and in fact the mouse mesenchymal stem cell also, possesses a bimolecular complex between CD44 and VLA4, such that if VLA4 gets triggered by CD44 binding, it becomes activated. So when we enforce E-selectin binding on the CD44 scaffold, and E-selectin binds to that H-cell molecule, that CD44 glycoform, it effectively activates integrins, importantly integrin VLA4, without chemokine input. This process we call the step 2 chemokine bypass pathway. We've worked out all of the molecular basis of this pathway, which is shown here in this cartoon. For those that are interested in this important biological nuance of the multi-step paradigm, I refer to you to a journal publication in the Proceedings of National Academy of Sciences of 2011, where we detail all the molecular processes involved in the binding of H-cell to E-selectin and the subsequent RAC-RAP activation of VLA4, such that the cell will then bind to VCAM and also fibronectin and transmigrate into the marrow parenchyma. So more broadly, this biology leads to an important revision of the multi-step paradigm. Enforced H-cell expression bypasses the need for chemokine input for transmigration of a cell in any endothelial bed that expresses both E-selectin and VCAM1. This pathway, this change in the multi-step paradigm, is now called the step 2 chemokine bypass pathway of cell migration. In a general sense, inflammatory cytokines TNF-alpha and IL-1 upregulate both E-selectin and VCAM-1 at sites of inflammation, at all endothelial beds of an injured tissue. So therefore, inflammation locks and loads recruitment of cells that express E-selectin ligands and VLA4. So therefore, the biology of H-cell expression, in particular enforced H-cell expression on mesenchymal stem cells or any other stem cell, if that cell expresses also VLA4, will direct trafficking of that cell to any site of tissue injury. So, shown here then is just a review statement that E-selectin VCAM-1 is a beacon for recruitment of H-cell positive, VLA4 positive cells to any site of tissue injury. So lastly, I want to emphasize what this whole biology and chemistry comes to. It is really the field of translational glycobiology. We are seeking to create the generations that follow in our footsteps of translational glycobiologists. We've created a training program specifically towards this goal. If you look here at these Venn diagrams, at the top are clinicians, physicians, surgeons, clinical researchers, etc. These individuals lack the informational basis of carbohydrates, of glycoscience, and the speed of medicine, in truth, is so rapid these days that it would not be possible for a clinician to literally understand glycoscience without the possibility of entering into a laboratory to get into the basic sciences. However, glycosciences is comprised of glycochemists and glycobiologists. The glycochemists are the organic chemists, the structural chemists, chemical engineers, the enzymologists, and the glycobiologists are immunologists, cancer biologists, microbiologists, cell biologists, all forms of biologic inquiry that also intersect glycoscience. So we feel that by training the glycobiologists and the glycochemists on relevant clinical principles, we'll be able to create translational glycobiologists. So rather than focusing on turning clinicians into glycoscientists, we're focused on turning glycoscientists into understandings to people who understand clinical medicine and understanding the human condition. And that is the essence of this training program. The training program is supported by the National Heart, Lung, Blood Institute. It's called a Program of Excellence in Glycosciences. It's supported by a program project grant. And the overarching clinical goal is to improve therapeutic outcomes for patients suffering from myelosuppressive conditions, bone marrow failure states and conditions, and myeloproliferative disorders by glycan engineering of hematopoietic cells. There are 3 projects, all linked together by an analytical core run by Vern Reinhold. I'm the PI of Project 1, which is the structure function analysis of glycans in early hematopoiesis. Project 2 is operated by Dr. Joseph Lau at the Roswell Park Cancer Institute. He's examining the regulation of glycan biosynthesis in hematopoiesis. And Project 3 is directed by Karen Hofmeister, a colleague at the Brigham Women's Hospital who's studying the structure, function, biology, and chemistry of glycans in thrombopoiesis. All projects are critically evaluating a clinically relevant problem using glycoscience-based approaches, translating these discoveries to the care of patients for diagnostic and clinical applications. And this translational-based training program is developing a new generation of translational glycobiologists. And I ask all of you that are looking at this lecture to consider joining us in this effort. So I want to thank the folks in the laboratory that made this work possible and my colleagues. This is our laboratory crest. It actually is not whimsical. It's produced for a purpose. Over here in the middle is a bird of prey. In this case, it's the hawk. A hawk will never lose sight of its prey. We seek to focus our efforts on a particular clinical problem and never lose sight of that problem, to keep ourselves focused on finding the answers in glycoscience-based approaches to treat the human condition. Over here on the left side of the shield is the sign of the caduceus, the sign of medicine. On the right is the sign of research. And at the middle here is the shield of the Harvard Medical School. At the top is written the word Fanaticus Pro Humanis et Clementia. So we are committed to humanity and mercy. That's the purpose of our research. And on the right here, a variety of very talented folks that have helped in this effort, a group of very talented technicians, some of whom have gone on to graduate training at this point, in particular Derek Kane and Taylor Schreiber. Derek Kane is now a PhD in immunology. Taylor Schreiber is now an MD PhD in immunology. A group of very talented postdocs. In particular, I want to highlight Dr. Jasmine Mirzaban and Dr. Nilesh Dagia and Dr. Sai Thankamoni. And colleagues at the Harvard Medical School that performed intravital microscopy, Joel Spencer and Charles Lin, and Roland Volgemuth, a scientist at Sigma, who helped us in creating the glycosyltransferases to perform the GPS reaction. All of this was supported by NIH, National Institutes of Health grants, and in particular the National Heart, Lung, Blood Institute, which has allowed us to obtain this program of excellence in glycoscience to continue these studies and train the future generation of translational glycobiologists. Thank you very much for your attention.